Hi, this is a science hangout from Science Mission. Today we have Andrea Globa from the University of British Columbia with us. She works in the lab of Dr. Shernaz Bamji, and she has won several awards in her short research career. She won a Banting and Best Canada Award in her master's and graduate school. Trainee Personal Development Award. She also has won several Best Poster Awards as well as the Best Grad Student Talk also. Welcome, Miss Andrea Globa.
Hi, nice to be here.
So thank you for joining us on Science Hangout. And let's talk about your background. Tell us about your background, education, etc.
Definitely. I did my undergraduate degree at the University of Winnipeg in biopsychology. And during my undergraduate training, I had the opportunity to do some research in some psychology labs and as well in some developmental biology labs. And I really enjoyed both fields of research, but I found that I was always asking the question, what is happening at the cell and protein level that's affecting behavior? And neither neither style of research really allowed me to answer those types of questions. And that's how I kind of got interested in pursuing research in neuroscience. And so in 2011, I came to the University of British Columbia and started working in Dr. Bamji's lab, looking at the effects of certain proteins on synapses and synaptic plasticity and how that affects learning and memory.
And I know you are a busy person with grad students, you need to work a lot of hours and all. So what are your other hobbies or interests other than research?
Well, I like to stay active, and living in Vancouver is a great city to do that because there's mountains and ocean really nearby. So my friends and I like to go hiking sometimes on the weekends, and in the summer it's nice to get outside at the beach or go kayaking. And I also have been a competitive softball player for a number of years, and I've recently picked that up again in Vancouver. I used to play a lot in Winnipeg, and now I'm playing on a team here.
And I saw your recent publication in Nature Neuroscience that is pretty interesting. So can you please talk and more about it and give your presentation on the work you have done?
Sure, yeah, that'll be the focus of my presentation. So do I just share the screen with you now?
Yeah, sure.
Okay. Okay. All right, so can you guys see it?
Yeah.
Okay, good. All right, so I'll be taking you through the findings of this paper today, but first, to give you some background, many researchers think that the cellular basis for addiction is synaptic plasticity gone awry in the dopaminergic circuitry of the brain. And we all know intuitively that the predisposition for addiction can vary from person to person. And it's thought that these differences are due to differences in genetic variation and certain single nucleotide polymorphisms in genes have been identified. And in particular, one of the families of genes that's been shown to have mutations in people that are at risk for addiction is cell adhesion molecules. And these are really important, some of them in synapse formation and some of them in synaptic plasticity. And so we were interested in looking at cell adhesion molecules and determining whether they play any role in the pathological plasticity that's been shown to occur in addiction. So to give you some background on the cell adhesion molecules in the brain, as I mentioned, there's several different types of these cell adhesion molecules. Now, most textbooks show the pre- and postsynaptic cleft hovering in space, but there are actually several families of cell adhesion proteins that hold these synaptic sites together. And some of these are important for synapse formation, and we were interested in cadherins, which are important for synaptic plasticity in particular. Now, cadherins are homophilic adhesion proteins that are present at both the pre- and postsynaptic membrane of synapses, and they're shown here in green. Now, cadherins interact both in cis and in trans and form a lattice that holds the pre- and postsynaptic terminals together. And these have been shown to play an important role in synaptic plasticity in the hippocampus. Increased cell adhesion is necessary for the strengthening of synapses or long-term potentiation. Now, after high-frequency stimulation or a learning event, there's increased cis clustering of cadherins at the membrane, as well as increases in the amount of both cadherin and AMPA receptors at the synapse, which results in the increase in size and stability of dendritic spines. So this increased cadherin adhesion is necessary for synapse strengthening. In fact, blocking cadherin interactions in the hippocampus can prevent both the strengthening of synapses and context-dependent fear conditioning. So we wanted to know if these same mechanisms of learning were important in plasticity in another region of the brain and in another type of behavior, in particular in addiction. So we decided to, before I continue, I just wanted to mention that some of the SNPs that I was talking about have been identified in cadherin proteins and some of their binding partners. And there's actually been shown to be increases in the expression of one of the binding partners, beta-catenin, after exposure to cocaine. So there was some backup, some evidence to support these ideas that we were going to follow up on. So we decided to look at a region of the brain that's involved in addiction. And so, as I mentioned, there's evidence from these GWAS studies that cadherins are important in plasticity. So first we looked at the ventral tegmental area, or VTA. Now, the VTA contains a population of dopamine cells, which project to the nucleus accumbens and the prefrontal cortex. And importantly, all addictive drugs increase dopamine transmission from the VTA to other parts of the brain. And these dopamine cells receive glutamatergic inputs and GABAergic inputs. And it's thought that by changing the strength of these inputs, you might be able to control the excitability of these dopamine cells and potentially drive changes that result in addiction. And so other groups have identified changes at synapses onto dopamine neurons in response to cocaine. So this is an excitatory synapse forming onto a dopamine neuron. And at baseline, the AMPA receptors at these synapses are GluA1,2 heteromers, and these allow sodium into the cell. However, after exposure to cocaine, there's this switch that occurs where GluA1,2 heteromers are removed from the synapse and GluA1 homomers are inserted. And these GluA1 homomers allow calcium into the cell, which results in this increased potentiation of the synapse. And so I should just mention, we know that cadherins can interact with the AMPA receptors. And so this is another reason we thought maybe cadherins are important in this type of plasticity. But first we wanted to see and confirm that they were actually expressed in this region of the brain. So we did some immunostaining for a number of classical cadherins, and we saw that N-cadherin and several related cadherins are all expressed in these dopamine cells in the VTA. And we did some electrophysiology with our collaborators in Stephanie Borgland's lab at the University of Calgary. And her postdoc Schwei did a spike timing dependent plasticity experiment. And so normally they do this technique a lot in their lab, where they use this spike timing protocol to drive these increases in EPSP amplitude after the spike timing protocol. And so to determine whether cadherins were important in this type of plasticity, Schwei pre-treated some brain slices with a peptide that blocks cadherin-cadherin interactions. And we found that when we pre-treated the slices with this peptide, we were able to block this type of plasticity. And to confirm the specificity of the peptide, we also used a scrambled peptide and showed that we were able to induce normal increases in synaptic strength. So this showed us that cadherins were present in the VTA and were important for synaptic plasticity in this region. So we next wanted to know if there were changes in cadherin localization at VTA synapses after mice developed a preference for cocaine. So to look at this, we used an experiment called conditioned place preference. And so in conditioned place preference, we use a 3-chambered box where chambers A and chambers B are distinct and yet equally preferable. And we watch where the mice, so we put the mice in the chamber during habituation day, and we see that they spend about equal time in chambers A and B. We then proceed to a set of conditioning days where mice receive cocaine in one chamber and saline in the other. And after doing this for several days in a row, the mice learn to associate the rewarding effects of the drug with the context in which it was experienced. And we can test their preference for the cocaine by, again, putting them in the chamber and allowing them to choose where they spend their time. And we see that wild-type mice spend significantly more time in the drug-paired chamber after conditioning. So at this time point, we collected brain samples from the VTA, and we did a technique called immunoelectron microscopy, which I'll tell you more about in a minute. And we also took another cohort of mice and did the exact same experiment, but after test day, we kept putting them in the chamber each day and watching their preference for the drug-paired chamber get reduced over time. And so after they'd extinguished their preference, we also collected samples for immunoelectron microscopy. So this is the behavioral data from that type of experiment. And you can see that after conditioning, the cocaine-treated mice showed a significant increase in their preference for the drug-paired chamber, whereas saline control mice, which got saline on both sides, spend about the same amount of time in the chamber throughout the experiment. So we've taken our VTA samples from these brains, and we wanted to identify excitatory and inhibitory synapses which were forming onto dopamine neurons in the VTA. But this is a very heterogeneous brain region with dopamine cells, lots of GABA cells, and so we really wanted to nail down the type of synapses we were looking at. So we first wanted to identify dopamine cells, and so to do that, we immunostained for a marker of dopamine cells, specifically the dopamine transporter, DAT. And we looked for that in postsynaptic compartments, and that allowed us to identify dopamine cells that were receiving inputs. We then also wanted to identify excitatory and inhibitory synapses, and you can do this by morphology, but we also immunostained for a marker of excitatory synapses like PSC95, and then we also looked at Cadherin for inhibitory synapses. And this allowed us to distinguish between the excitatory and inhibitory inputs forming onto these dopamine cells. Now, the final marker we looked at was for cadherin. And so when we immunostained for cadherin, which is the smallest bead size here with the blue arrows, we see this distribution around synapses. And the way that we quantified this was we measured the distance of each of these immunogold beads to the pre- or postsynaptic membrane, depending on which compartment they were in. And this really gives us a sense of the distribution of the protein around the synapse. And just to kind of let you guys know, we looked at the size of, based on the crystal structure of the size of the protein and the known sizes of antibodies and the gold beads, we considered any labeling within 40 nanometers to be labeling cadherin that is likely inserted in the synaptic membrane and participating in transsynaptic adhesion. So when we did this for the mice that went through our behavioral task, we saw that the cocaine group showed the significant increase in the amount of cadherin localized to the synaptic membrane compared to their saline controls. And this was strongly correlated with their behavioral data, such that the mice that spent the most time in the conditioned chamber had the greatest amount of cadherin at the synaptic membrane. Now, in contrast, when we looked at inhibitory synapses, we didn't see any changes, suggesting that these changes in cadherin localization were really specific to excitatory inputs. And so we did a number of control experiments. As I mentioned before, we looked at what happens after extinction and showed that so the mice had developed this preference for cocaine. We showed this increase in cadherin at the membrane, but after they extinguished their preference, the amount of cadherin at the membrane was back at baseline. We also wanted to know whether the changes we saw were purely due to the physiological effects of cocaine, or whether this would occur due to, whether this was due to the conditioning and that associative learning that was occurring. And when we looked at a home cage control group of mice, which received cocaine on the same schedule as our behavioral group, but didn't go in any novel environment, they just stayed in their home cage each day, we showed that they didn't have an increase in the cadherin localized to the membrane, which suggested to us that there really was something about that associative learning that was driving these changes. And so, and to sort of look at the extinction and as a control for the extinction experiment, we also took a group of mice where we conditioned them in the novel context of the conditioning chamber. And then rather than extinguishing them in the chamber each day, we kept them in their home cage for 6 more days to see whether the effects that we saw at the extinction time point were due to that unlearning, or some would argue is a new form of learning that the chamber is no longer preferable, or whether this was a passive effect that would happen no matter what. And what we saw was that after the home cage extinction is what we kind of called it when we were doing the experiment. After that, 6 days of just staying in the home cage, cadherin levels were back at baseline as well, which suggested that there needed to be this transient increase in cadherin during conditioning, but this wasn't necessary for extinction. This was going to be reduced no matter what. And then finally, we did a food CPP experiment. So we wanted to know whether the effects we saw were specific to addictive drugs or whether any natural reward would also drive changes. And so although these mice showed a conditioning effect where they did show preference for the bacon-flavored treats that we conditioned them with in the one chamber, they didn't show an increase in cadherin localization. So we also looked at GLUA1 localization at synapses, and similarly to cadherin, we saw that after conditioning in the drug-paired chamber, there was a significant increase in GLUA1 localized to the membrane, which was also strongly correlated with each mouse's behavior. And so we wanted to know if cadherin and GLUA1 were trafficking together, and one other experiment we did was we immunostained for GLUA1 and cadherin in the same samples, and we showed that synapses that were high in GLUA1 were also high in cadherin in both our saline controls and in our cocaine CPP mice. So this suggested to us that the cadherin and GLUA1 were trafficking together, whether we were looking at saline-treated mice or cocaine-treated mice. But the difference we saw was in the number of synapses that were fit into each of these categories. So the cocaine group had a significantly higher number of synapses that were both high in cadherin and GLUA1, and a significantly fewer synapses that were low in both cadherin and GLUA1, suggesting that more cadherin and GLUA1 were both trafficking to these synapses. So we'd shown that there are these changes in cadherin and GLUA1 localization after mice develop a preference for cocaine. So we next wanted to know if we stabilize cadherin at the membrane, could we have any effect on the drug-mediated learning or the drug-mediated behavior? And so to examine this, we took a transgenic approach where we used a conditionally stabilized beta-catenin mouse line. And so these mice have a floxed exon 3 allele. And exon 3 contains some phosphorylation sites which, when they're phosphorylated, normally mark beta-catenin for degradation by its normal mechanisms. But in these mice, we were able to produce a mutant form of beta-catenin, which could not be phosphorylated and therefore could not be broken down. And so we used a Cre line where we were only expressing Cre in dopamine cells, which meant that we were increasing beta-catenin stability specifically in dopamine cells. And so to show the validation of that, you can see that we see significantly higher levels of beta-catenin immunostaining in our mutant mice compared to our wild-type controls. And so by increasing beta-catenin stability, we could increase cadherin's stabilization at the synaptic membrane. Beta-catenin interacts with cadherin and prevents it from being endocytosed when it's interacting. So by increasing the amount of beta-catenin in these dopamine cells, we were increasing cadherin's stability at the membrane. And for any of you cell and developmental biologists out there who are listening, we did look at Wnt signaling and confirmed that in our mice, we didn't see any increases in the expression of Wnt targets in these dopamine cells. Furthermore, our lab has used this mouse line in the past in the hippocampus, and we've shown that there weren't off-target downstream effects on Wnt signaling in these mice. So just to let you guys know in anticipation of a question. So when we took these mice and we took them through the same conditioning condition place preference experiment, we showed that they spent significantly less time in the conditioned chamber compared to our wild-type control mice. And so this was really interesting to us. And just to remind you guys how the plasticity in this part of the brain works, after exposure to cocaine or other drugs of abuse, at these excitatory inputs onto dopamine cells undergo this change where normally they're GLUA1-2 heteromers, but after cocaine administration, we get the insertion of more GLUA1 homomers at the synapse. And what's interesting is one paper has shown that the internalization of the GLUA1-2 heteromers is necessary for this type of plasticity. And if you block that internalization, you actually block the insertion of GLUA1 homomers. And so we thought that was pretty interesting, and we thought that perhaps cadherins were impairing plasticity in this region of the brain by interfering with this AMPA receptor cycling. Oh, oops. And so to test this, we went back to our immuno-EM, and we took immuno-EM samples from wild type and the beta-catenin mutant mice, and we did some more immuno-EM. And so as we've seen previously in our wild type mice, after conditioning, they show this increase in cadherin localized to the synaptic membrane. But in our mutant mice, there was an increase in cadherin at baseline, and then there was no further increase after conditioning. And so this fit really well with our expectation that these mutants would have increased cadherin stability at the membrane. And it was interesting to see that there was no further change after the conditioning. We then looked at GluA2 localization. And so in our wild type mice, we saw that after conditioning, there was this significant decrease in the amount of GluA2 localized to the synaptic membrane, which indicated to us that those GluA1-2 heteromers were likely being removed from the synapse in these wild type mice. But in our mutant mice, this was completely blocked, which was indicating to us that perhaps cadherin was interfering with this type of plasticity. Finally, we looked at GluA1 localization, and as we've seen previously in our wild-type mice, we show this significant increase in the amount of GluA1 at the synaptic membrane after conditioning, indicating to us that GluA1 homomers were being inserted at the synaptic membrane. But again, in our mutant mice, this was completely blocked, suggesting to us that by blocking the internalization of the GluA1-2 heteromers, we were likely interfering with the insertion of GluA1 homomers at the synapse. So we also examined this using electrophysiology, again with Stephanie Borgland's lab at the University of Calgary. So her postdoc Schwai looked at a few different measures that are commonly used to look at the type of receptors present at these excitatory synapses onto dopamine cells in the VTA. So first they looked at something called the AMPA to NMDA receptor ratio. And in the wild-type mice, they saw that after a cocaine injection, there was this increase in the AN ratio, which others have shown indicates that there are more AMPA receptors being inserted at these synapses. But in our mutant mice, again, this was completely blocked, indicating again that the increased cadherin was interfering with plasticity in this brain region. And they also looked at something called the rectification index. So this gives us an idea of the type of AMPA receptor that's present at these synapses. Now, GluA1-2 heteromer type AMPA receptors, as I mentioned, are calcium impermeable, and therefore they have a linear rectification index. But the GluA1 homomer type AMPA receptors are inwardly rectifying, and this results in an increase in the rectification index, which is shown here. And this is what we saw in our wild-type animals. And there's an increase in linear inwardly rectifying synapses. Now, in our mutant animals, again, this was completely blocked, indicating that the GluA1 homomers were not being inserted at these synapses. So together, this data shows that there are impairments in plasticity in our beta-catenin stabilized mice. So to summarize, normally after mice are given cocaine, paired with a novel environment, there's a removal of the GluA1-2 heteromers and insertion of GluA1 homomers at these synapses. And this change in AMPA receptor subtype is facilitated by cadherin turnover and increased cadherin stability at the synaptic membrane, which results in the potentiation of synapses and a strong preference for cocaine. But in our mutant mice, where cadherin is already stable at the synaptic membrane, the internalization of GluA1-2 heteromers is impaired. And some researchers believe that there's only a certain number of AMPA receptor slots at these synapses. So when the GluA1-2 heteromer internalization is blocked, there's no space for other receptors. And the insertion of these calcium-permeable GluA1 homomers is also blocked. So this results in impairments in synaptic plasticity and impairments in the acquisition of conditioned place preference. So that kind of sums up the whole paper. We're kind of interested we've talked a lot about what the next step for this project might be. And we're interested in looking at other targets that can interfere with or facilitate cadherin adhesion and might play a role in this type of plasticity. And hopefully we could identify proteins that are more specific to this circuit and this pathway. mentioned cadherins are also very important in learning and important in plasticity and other brain regions. So ideally, it'd be interesting to discover some protein that is more specifically expressed in the midbrain or more specifically expressed in dopamine cells that could act on cadherins. So I just want to take a moment to thank all the lab members that I worked with, specifically with my co-first author, Fergal Mills, who was a huge part of this project, and as well the funding agencies like CIHR, which supported myself and the lab. All right.
That's a good presentation, Andrea. Can you please?
Yeah.
Yeah. Thank you so much. It was very interesting.
Thanks.
I have a couple of questions.
Sure.
Like, have you looked at any protein synthesis, or there is no protein synthesis effect on the changing of these?
We didn't examine that specifically. We did look at the total labeling of cadherins when we did the immuno-EM experiments, and we didn't see any changes in the levels when we looked at the EM level. But we haven't examined it by Western blots or anything like that. The VTA of the mice are very small, so we'd need to pool the protein from several mice to do that type of experiment properly.
Okay. And is there any other glutamate receptors, AMPA receptors have several subtypes, right?
Yeah,
so
we were looking mainly at GLUA1 and GLUA2. I have been asked whether GLUA3 is part of the AMPA receptors that we were looking at. We didn't specifically immunostain for GLUA3, although I guess it could definitely be part of some of those AMPA receptor subtypes.
And do you know where exactly on the cadherin and the receptor interaction that happens? What is the on the AMPA receptor?
Well, cadherin has been shown to interact with both GLUA1 and GLUA2, both directly and indirectly through proteins like GRIP. And so it's because that was something we were wondering about as well. In the first part of the talk, I told you about how cadherins are important for driving synaptic plasticity. And then I've shown when you increase cadherin, we impair plasticity. So it's kind of a bit counterintuitive at first. But I think what's really important is the timing of when you increase the cadherin localized to the membrane. So in the wild-type mice, cadherin increases during potentiation when those GLUA1 homomers are present at the synapse. So I think then it will help stabilize the GLUA1 homomers and drive that plasticity. Whereas in our mutant mice, we have increased cadherin at baseline. And so at baseline, we have the GLUA1-2 heteromers present, and therefore we're stabilizing the receptors that are going to be calcium impermeable, and that's where we're seeing the impairments in plasticity and in behavior.
Okay. And have you used any other drugs? This is you have done cocaine, CPP, and all
We just did a cocaine CPP. Other groups that have looked at this type of plasticity have shown that other drugs such as amphetamines and nicotine and ethanol all drive the same type of changes at these synapses. So I would imagine we might see similar effects with other drugs, but that's not something we've looked at directly.
It looks like it is a common mechanism, so you can, universally, you can, if you think that, okay, for some way of intervention, any other drug also you could use the same sort of intervention for.
Yeah, I mean, hopefully there's some commonality. I mean, it definitely would be something to look at, and obviously all these drugs act slightly differently, but they do seem to drive some similar changes in plasticity. So. Okay,
so what do you want to do with this current project, going forward? So are you going to work on that more, or how does, what is your plan?
Well, personally, my plan is to graduate soon. So there will be a few, a new postdoc who's looking at some of the same things, looking at some other proteins. Our lab has previously shown that enzymes like DHHC5 can act on the cadherin adhesion complex by palmitoylating delta-catenin, which is another binding protein of cadherin. So it'd be interesting to see if DHHC5 expression might have effects on this type of plasticity. So those sort of avenues, like seeing what other downstream proteins might have an effect on cadherin adhesion in the VTA. I also think it would be interesting to look at plasticity in other related brain regions, such as the nucleus accumbens, because we know that this type of plasticity in the VTA is quite transient and drives downstream changes in the nucleus accumbens. So it would be interesting to look at that brain region and see if what type of changes in cadherins or other cell adhesion proteins might be occurring there.
Yeah, that's an interesting question in the sense that addiction is a long term. I mean, like once you get addicted, like some changes has to happen for a longer period of time. So certainly there might be more to it than
definitely. And we are not trying to say that conditioning with 3 doses of cocaine is equivalent to a lifetime of drug addiction and those types of other factors that might be at play for sure. But yeah, it's definitely, you know, it's a model, and you have to start with something to work from.
It's an interesting finding, really. Like when I looked at news about that, so I was really intrigued that like if you can intervene some way, like that would be. I mean, like that is one of the biggest challenges what we have is the drug abuse these days. Have some understand more about what happens in the brain, so there is a possibility of intervening in some form. Anyway, thank you so much. I really appreciate you taking time, and good luck with your defending thesis in the summer and for all your future research endeavors. I really appreciate your time.
Thanks very much. It's been fun.
Thank you.